Immunology, cytokines. All right, so cytokines, once again, are going to help us kind of mediate our immune response in certain ways. So there's certain types of cytokines that matter in different ways. Once again, I'll tell you the mnemonic hot T-bone steak is very, very helpful. We'll start off with that. IL-1 is for fever, which stands for the hot. IL-2 is for the T, which stimulates T cells. IL-3 is for the bone because it stimulates bone marrow. IL-4 is for the E in steak, and it stimulates IgE production. IL-5 stimulates the IgA production, the A in steak. And the K in steak is for IL-6, which stimulates acute, K for acute phase protein reactants or products. All right, so that's the first thing that we kind of have to take away from cytokines. It's just a quick overview of what goes on, but let's kind of dive into it a little bit deeper. So these are all secreted by macrophages: IL-1, IL-6, TNF-alpha, IL-8, and IL-12. So IL-1, secreted by macrophage, is going to cause fever, and it's going to help with acute inflammation. All right, it's essentially itself an acute phase reactant, IL-1. All right. IL-6, secreted by macrophages as well, is going to be for acute phase proteins, okay? So same thing, it stimulates production of acute phase proteins though. That's the largest yield thing to know for IL-6. TNF-alpha is also produced by macrophages. Now, what it does specifically is that it's going to cause white blood cell recruitment and vascular leak, okay? So it's important in the pathogenesis of septic shock. But what the key thing here that you need to know is this: it maintains granulomas in TB. So TNF-alphas maintain granulomas in TB, okay? Now, IL-1, IL-6, and TNF-alpha are our main mediators of fever and sepsis. So these are our acute phase kind of reactants. So that's just kind of the end-all be-all for IL-1, IL-6, and TNF-alpha. But remember, IL-1 mainly for fever, IL-6 mainly for acute phase reactants, and TNF-alpha, what I want you to mainly remember, is for the maintenance of the granulomas in TB. Next, also released by macrophages, is IL-8. IL-8 is used for chemotactic factor for neutrophils. So it helps for chemotaxis, right? Remember, what are the causes that help recruit neutrophils to certain sites? One is going to be IL-8, as we discussed just now, which are produced by macrophages. The second one's going to be a complement, C5a. And the last is going to be LTB4, okay? Leukotriene B4. These are all recruit neutrophils. Last secretion of macrophages is going to be IL-12. What IL-12 specifically does is going to facilitate granuloma formation in TB, okay? It's going to activate our T helper cells into Th1 cells. That's very key. So T helper cells are going to be activated into the class of Th1 cells, all right? Next is secreted by T cells. So what's specifically secreted by T cells are IL-2 and our IL-3. IL-2 is going to help stimulate growth of T cells. So all T cells, helper T cells, cytotoxic T cells, regulatory T cells, and NK cells. So IL-2 is just for help of production of more T cells. IL-3 is to help in production of bone marrow stem cells. All right, not too high yield for IL-3. Now, from Th1 cells, so our T cells now have our T helper cells have now produced and become a different cell line, Th1 cells and Th2 cells. Those are the two cell lines you have to know. Th1 cell lines are going to produce interferon gamma, so IFN gamma. And what these do are they're going to be essentially stimulating macrophages to kill phagocytic pathogens, okay? So that's the whole entire point, is IFN gamma is going to recruit macrophages in order to kill via phagocytosis. Okay, so it's going to activate macrophages to induce granuloma formation. That's what IFN gamma does. It's produced by Th1 cells. Next is Th2 cells. They produce four things: IFN or interleukin 4. Interleukin 4, remember, is associated with stimulation of IgE production in response to, can you guess? Exactly, eosinophil production against parasites or anaphylactic reaction. Okay, it also promotes growth of B cells. Now, interleukin 5, IL-5, is going to promote growth of indifferentiation of B cells as well, but it's going to enhance class switching to IgA, okay? And it's going to help differentiate eosinophils, all right? So keep that in the back of your mind, although it can be confusing. Remember, IL-4 only acts with eosinophils because it produces IgE, whereas IL-5 actually helps stimulate growth and differentiation of eosinophils, okay? Next is IL-10. IL-10 and TGF-beta, remember, are both going to attenuate the immune response. So it's going to help decrease expression of our MHC class 2s, and it's just going to relax the system by secreting regulatory T cells. Lastly is IL-13, very low yield. It produces and promotes IgE production, okay? Now, kind of the main takeaway that I want you to know is, of course, how granulomas are formed. Okay, so this is kind of the main takeaway of this whole entire page. I want you to remember hot T-bone steak, obviously, and I want you to remember what helps with chemotaxis of neutrophils. But what creates granulomas is very high yield. So let's start with macrophages. So macrophages are recruited to a site because they sense that there's an antigen, a foreign antigen. So macrophages sense that there is a foreign antigen, and what they're going to do is that they're going to produce IL-12, okay, in response to the foreign antigen. And what IL-12 goes and does is that it's going to differentiate T helper cells into Th1 class cells, T helper cells. So T helper cells are going to turn into T helper 1 cells. All right, so that's where it's going to differentiate. T helper 1 cells are going to produce two things. They're going to produce IL-2, which makes more T helper 1 cells. And they're also going to produce IFN gamma, which recruits more macrophages. Okay? And then those macrophages that are being recruited again are going to produce TNF alpha, which maintains the granuloma. So this is what I want you to write down right now. Write macrophages and put an arrow towards IL-12. Then from IL-12, I want you to put a down arrow, okay, which shows T helper cells differentiating into T helper 1 cells. Then the next arrow I want you to write is a circular arrow. So the arrow is going to start at T helper 1 cells, and then it's going to finish right back at T helper 1 cells, a circular arrow. And I'm going to write IL-2 to show you that T helper 1 cells produce IL-2, which helps produce more T helper 1 cells. Also from T helper 1 cells, I want you to write an arrow to IFN gamma. From IFN gamma, write an arrow to macrophages. Just write macrophages again. And then from macrophages again, write TNF alpha. And then that's going to maintain your granuloma. All right, so that's the whole process about the cytokines. Granuloma formation is probably the highest yield about what you need to know about the cytokines, but that's all.